Hello and welcome to Unacademy, a one-stop destination for all English medium UPSC aspirants. Welcome to the new initiative Conquer Mains, the Quality Enrichment Program for Mains 2024. Today is the first session in Science and Technology. But before we proceed with the session, let me introduce about the Conquer Mains. The program includes a total of 60 sessions, 90 hours of discussion and 5 topics in each session, and hence the program includes a total of 300 topics from current affairs and the connected static portion in all the GS subjects from GS paper 1, paper 2 and paper 3. In each session, the subject specific expert faculty will be covering 5 most important topics. For one of these topics, a question and the model answer will be provided during the session. For the remaining topics, we conduct weekly tests on Sundays. Your answers will be evaluated and a detailed feedback will be provided to you. Along with that, we also provide a comprehensive notes for all the topics discussed during the program. The first session in all the subjects is offered free of cost on our YouTube channel Unacademy IAS English. With this, let us continue our discussion with science and technology. As planned, in today's session we will be covering 5 topics, and the 5 topics are Aditya L1, its payloads and its objectives, genomics and its applications, Agni 5, the intercontinental ballistic missile with MIRV technology, sodium ion batteries, in what way they are better when compared to the lithium ion batteries and why India needs sodium ion batteries, and finally gene therapy and its applications. Let us start our discussion with Aditya L1. Aditya L1 is India's first space-based solar observatory. Means it is located in the space. It will be orbiting around the sun, constantly observing the sun and imaging the sun. And Aditya L1 was named earlier as Aditya 1, but it is renamed as Aditya L1. In the name, L1 stands for the Lagrange point number 1. Let us try to understand what is Lagrange point. It is a point where the gravitational force of 2 large bodies and the velocity of the spacecraft balance each other. Lagrange point is a point where 3 forces balance each other: the gravitational force of 2 large bodies and the velocity of the moving satellite. If we try to understand this in the context of Aditya L1, Aditya L1 is located here. At this point, it is balanced by the gravitational force of the sun, the gravitational force of the earth and the velocity of Aditya L1. These 3 forces balance each other so that Aditya L1 will be stabilized at this point. We don't need to spend fuel to maintain this particular position of Aditya L1 because it is maintained by the balance of these 3 forces. Such points are called as Lagrange points. In the Sun and Earth system, we have a total of 5 Lagrange points. The Lagrange point number 1 where the Aditya L1 is located, and the Lagrange point number 2 where the James Webb Space Telescope is located. Lagrange point number 3 here, L4 here and L5 here. And Aditya L1 is placed at the Lagrange point number 1. And Aditya L1 was launched in the rocket called PSLV XL in the mission PSLV C-57. And what are the objectives of Aditya L1? Aditya L1 can analyze the solar winds, solar flares, pre-flare activities, dynamics of the space weather, that is how the space weather is changing, propagation of particles, coronal mass ejections, solar storms, etc. There is a lot of terminology here. To properly understand the objectives of Aditya L1, we should first get familiarized with these terms. Let us try to understand these terms and then go back to our discussion. Firstly, what do you mean by the sun's atmosphere? Sun's atmosphere is divided into 3 layers. The topmost layer of the sun's atmosphere is corona, and below the corona comes chromosphere, and below that, that is the innermost layer of the sun's atmosphere, is photosphere. With that, the atmosphere of the sun ends. And after that, there are 3 more layers called the convective zone, radiative zone, and the central most part of the sun, that is core. So total 6 layers from the periphery to the center: corona, chromosphere, photosphere, convective zone, radiative zone, and then the core. These are the layers of the sun. And then, what do you mean by solar flares? From the sun's atmosphere, if high energy electromagnetic radiations like X-rays, gamma rays are released, that is called as solar flare. So solar flare is the electromagnetic waves or the radiation released from the sun's atmosphere. Next, what is solar wind? From the sun's atmosphere, if high energy particles are released, like for example, the particles like electrons, protons, and alpha particles. So what is the difference between solar flares and solar wind? Solar flares are the electromagnetic radiations like X-rays, gamma rays, and solar wind is high energy particles like electrons, protons, and alpha particles. Along with these two, we should also know another phenomenon that is called coronal mass ejection. If you observe this word, coronal mass ejection, it means some mass, means matter, is ejected, means released from the corona, that is the outermost layer of the sun's atmosphere. When some mass or matter is released from the sun's corona, that is called coronal mass ejection. This matter is in the state of plasma. As we all know, we have solid, liquid, vapor, different states of matter, but this mass is ejected in the state of plasma. It means when the plasma is released from the sun's corona, that is called coronal mass ejection. And the next one is geomagnetic storm. Let us try to understand what is the geomagnetic storm. Let us imagine this is the sun. From the sun, particles means solar wind, okay, or radiations means solar flares, or plasma, that means coronal mass ejection, are coming out. while they are coming out from the sun's corona, they also drag the sun's magnetic field outside. The solar winds, flares and coronal mass ejections, they are dragging the sun's magnetic field towards the earth. We have the earth here, towards the earth. Then the sun's magnetic field will disturb the earth's magnetic field. That disturbance in the earth's magnetic field because of the dragged out sun's magnetic field is called as geomagnetic storm. We call that as geomagnetic storm. Then what is a solar storm? Solar storm is similar to the geomagnetic storm. The only difference is the solar storm lasts only for few minutes to few hours, but geomagnetic storms will linger on the earth for a few days to few weeks also. It means less intense geomagnetic storms can be called as solar storms. Okay. Why did we discuss about solar wind, solar flare, coronal mass ejection, geomagnetic storm and solar storms? Because the objective of Aditya L1 is to study about all these things. Because these geomagnetic storms will disturb the GPS navigation, our Wi-Fi technology, our satellite communication and everything related to either electricity or magnetism or electromagnetic radiations. So, we should keep a track of these particular emissions from the sun. Constantly observing the sun and tracking these emissions is the important objective of Aditya L1. Now, how does Aditya L1 observe all these things? To observe all these things, it requires certain instruments, and we call them as payloads. One important payload is a spectroscope. Remember the word spectroscope. First, spectroscope means what? In this, it is an instrument that will split the light into different wavelengths or frequencies and will only observe the light. I repeat, this instrument will split the light waves into different wavelengths or frequencies and will observe those waves. Just observe means it is not measuring it. There is another type of instrument called spectrometer. From the name itself, it is evident that we are using the word meter. Means it can also measure. It means this instrument not only observes the light or electromagnetic radiation in different wavelengths, but it can also measure the intensity of the waves in each wavelength. It can also measure the intensity of the electromagnetic waves in different wavelengths or frequencies. Such an instrument is called as spectrometer. And in our discussion, another term also comes, that is hard X-rays and soft X-rays. What is the context of these two terms? Because Aditya L1 will be analyzing the hard X-rays and the soft X-rays which are released from the sun's corona as a part of the solar flares. What do you mean by hard X-rays? Simple. Hard X-rays are high frequency X-rays and they have more penetration capacity. Soft X-rays are slightly low frequency X-rays which have a comparatively less penetration capacity. As we completed understanding all the terminology, let us again proceed with the understand the objectives of Aditya L1. If you see the objectives, Aditya L1 can analyze solar winds, solar flares, pre-flare activity I mean, before the solar flare is released from the sun, are there any activities in the sun? Dynamics of the space weather, how the space weather is changing over a period of time. Propagation of the particles, that is in the solar winds, and fields, that is a magnetic field of the sun, how the sun's magnetic field is changing. Coronal mass ejections, solar storms, geomagnetic storms. Keeping an eye or watch on all these phenomena is the main objective of Aditya L1. To observe all these things, Aditya L1 is having a total of 7 payloads or 7 scientific instruments. In this, 4 payloads directly view the sun and 3 payloads carry out in situ study on the particles. In situ means at the site. These 3 instruments will collect the particles from the sun's corona or the coronal mass ejections and will analyze these particles at that site. I repeat, these 3 instruments will collect the particles from the coronal mass ejections or the corona and will analyze them at the site. Okay, in situ particle analysis and the analysis of the fields. What fields? Magnetic fields. So 4 instruments will be imaging the sun, 3 instruments will be working on the analysis of the particles at the site. Here is the list of the 7 payloads. The first payload is visible emission line coronagraph. What it will do? Its function is corona imaging and spectroscopy. We already understood the term spectroscopy. Spectroscopy is an instrument that will try to analyze the electromagnetic radiations in different wavelengths. And the second instrument is solar ultraviolet imaging telescope. Its objective is it will study the photosphere and chromosphere imaging. It will take the images of the second and third layers of the sun's atmosphere, not the corona, not the outermost layer. Outermost layer will be imaged by the visible emission line coronagraph and the second and third layers of the sun's atmosphere are imaged by the solar ultraviolet imaging telescope suite. The third instrument is solar low energy X-ray spectrometer. Low energy, you all know that energy and frequency are directly proportional. Low energy X-rays means low frequency X-rays, means soft X-rays. Solar low energy X-ray spectrometer will analyze the soft X-rays released from the sun's corona. And it is a spectrometer, means it will not only observe the X-rays, it can also measure, measurement. It can also measure the intensity of these X-rays. The next instrument is high energy L1 orbiting X-ray spectrometer. High energy means high frequency, means hard X-rays. It will observe, not only observe, it is a spectrometer, means it will also measure the intensity of the high energy or the hard X-rays released from the sun's corona. And we also have 3 more instruments in the Aditya L1. One is Aditya Solar Wind Particle Experiment. Solar Wind Particle Experiment. It will collect the particles from the solar wind. I hope you remember solar wind is about high energy particles like alpha particles, electrons, etc. It will analyze the particles in the solar wind. It will analyze the particles in the solar wind. Next one, plasma analyzer package for Aditya. Plasma we have in the solar wind and the coronal mass ejection. It will analyze the plasma in the solar wind and the coronal mass ejections. It is a particle analyzer. It will analyze what kind of particles are present and what proportion of these particles are present in the solar wind. Lastly, advanced triaxial high resolution digital magnetometers. Magnetometer means it will measure the intensity of the magnetic field, which is very important because the intensity of the sun's magnetic field which is dragged out, okay, will determine the intensity of the solar storm or the geomagnetic storm that the earth experiences. Okay, so it will do in situ magnetic field analysis. It will measure the intensity of the magnetic field. These are the objectives and the payloads of Aditya L1, and that's all about Aditya L1. Now moving on to one more topic. The next topic we are discussing is genomics and the applications of genomics. Before we proceed with the applications, let us try to understand what is genomics. For that we should first know what is a genome. Genome is one set of the total genetic material. One set of the total genetic material in one cell. As you all know, in the human beings, one cell contains a total of 46 chromosomes. In this, 23 chromosomes comes from the mother and the other 23 chromosomes comes from the father. So, in the human beings, one genome or one set of all the chromosomes means 23 chromosomes. That is a genome, one set of the total genetic content of a cell. It means one set of all chromosomes in a cell. That is 23 chromosomes together, we call it as one genome. And what do you mean by the word genomics? Genomics means is a study of genomes. Genomics is a study of the genomes. Now comes the question, why to study the genomes? What is the significance of genomics? And as I told you in the introduction of this Conquer Mains program, in this program, in each session, I will be giving you one question and the model answer for that. For the remaining topics, you will be getting a weekly test on the next Sunday. So, the model answer that I am providing is for this topic. The question is, discuss the role of genomics in the recent advancements in biotechnology. Enlist the recent initiatives in India towards the genomic research. What initiatives are taken in India towards the research in the field of genomics? This is the question. So for this, we have to know what is genomics, what are the new advancements in the field of genomics, and what are the new initiatives taken in India. So let us divide the answer into these three segments. As you all know, it is better we divide the answer into introduction, body and conclusion. It's not that you have to use the terms like introduction and conclusion, but you have to rightly introduce the topic about which you are writing. So here, as a part of the introduction, we are defining the term genome. Genome is the complete set of the genes or genetic material present in a cell. A genome in human beings is comprised of 23 chromosomes. And then we are defining the word genomics. As I already told you, genomics means the study of genomes. Genomics is a study of the genome of a person or an organism. It means we need not study the genome of a human being only. We can also study the genome of a bacteria or a virus or a plant or any organism to that matter. So genomics is the study of genome of an organism or a person or a human being. This completely is about the introduction. And then moving towards the body of the answer, the main content of the answer, that is, genomics plays an important role in the recent advancements in biotechnology. So let us try to understand what are the recent developments in the genomics in various fields. Firstly, the importance of genomics in agriculture. First, fast breeder varieties. How do we know which is a fast breeder variety? Let us suppose you have some 10 varieties of a plant. 10 varieties of a plant. Then what we do? We study the genomes of. We study the genomes of all the 10 varieties. And then we will try to understand which one of them will be a fast breeding variety, which one of them can give a quicker yield. How do we know which variety is a fast breeding variety? By studying about the genomes, because our genetic material will determine every aspect of our biological being. Like for example, in the human beings, our skin color, intelligence, height, nose shape are decided by the genome sequence, DNA sequence. Same way, when it comes to a plant, whether a particular plant variety is a fast breeding or slow breeding depends on the genome sequence. So take 10 varieties of that crop, analyze the genome sequence of all the 10 varieties and understand which of these 10 varieties will give the faster yield, that is a fast breeding variety. Then we can selectively breed that variety. Genomics plays a role in doing this, that is faster breeding varieties. Next one, plants' reaction to the biotic and abiotic stresses. What are the biotic stress that we have? What are the biotic stresses faced by the plant? Biotic stress is about the viruses, infection by the plant viruses or pest attack. These are called as the biotic stress. Infection by a plant virus or an attack by a pest are the biotic stresses. Then what are the abiotic stresses? That is high intensity light and very high temperature, or lesser availability of water, that is water scarcity or the drought. These are some abiotic stresses. How do you understand the mechanism of the plant's reaction to biotic and abiotic stress? It means when a pest attack, how the plant will respond. We can understand it by studying the genome of the plant. When you study the genome of the plant, you will understand how the plant is responding to this biotic and abiotic stresses. What is the advantage of that? Let us suppose you have two varieties of the plant, two different varieties of a plant. The first variety is more resistant to these biotic and abiotic stresses. The second variety B is less resistant to these biotic and abiotic stresses. Then we can choose to selectively breed the first variety. But how do you understand which variety will be more resistant to these stresses? By analyzing their genome sequences. Because genome sequence, that is a DNA sequence, will decide every biological characteristic of an organism. Its capability of pest resistance or its capability of drought resistance or its capability of how quickly it can breed. About all these things we can only understand by studying about the genome of that particular plant. It means genomics is very important in this field. Next, we can also understand about the mechanism of the pest attack. It means by studying the DNA, we can understand by what mechanism the pest is attacking the plant. If you know this, like say for example, there is a receptor. For example, this is a plant cell and there is a receptor, and the plant virus is binding to this receptor and getting entry into the plant cell through this receptor. How do you know about this receptor? By studying the genomics, because you have a gene for this particular receptor. This is the mechanism by which the virus is infecting the plant. If you know this, we can try to delete this receptor. It means gate is removed. When the gate is removed, then there is no more entry for the virus. I repeat, this is a plant cell and it is a receptor on which the virus will bind, and the virus is entering into the plant cell through this receptor. Now what we have done is we studied the genome of this plant very well and we understood where is the gene for this receptor and we deleted that gene. It means we do not have this receptor anymore, so the virus cannot infect the plant cell. So, by genomics, we can also understand the mechanism of the pest attack. By understanding the mechanism of the pest attack, we can prevent the pest attack. I mean, we can create pest-resistant varieties of these crops. Lastly, we can also understand the molecular mechanism of herbicide, drought and pest resistance. It means by what mechanism the plant is becoming resistant to the drought, herbicides and the pest. By observing this, we can try to create similar changes in the DNA of the other plants through genetic engineering, and we can try to make the other plants also pest resistant or drought resistant. I repeat, let us suppose one plant is herbicide resistant. Find which gene is responsible for the herbicide resistance, so that we can insert this gene in the other plant to make the other plant also herbicide resistant. This is the role of genomics in agriculture. Let us now try to understand the role of genomics in the health. First, understanding the mechanism of infection and diagnosis. Again, understanding the mechanism of infection. If you take this example of SARS-CoV-2, let us suppose this is a human cell and you have a receptor called as ACE2 receptor. This is like a gate for the SARS coronavirus 2, that is the virus that will cause the COVID-19 infection, and the virus spike protein will bind to this ACE2 receptor. Okay, and what we do here is we do the genomics. I mean, we study about the genome. By studying about the genome, we will try to identify, we can try to identify the gene for ACE2, and then we can try to suppress. We can try to suppress ACE2. Then the coronavirus cannot enter into the human cell. Let us take one more such example. Let us suppose this is the human cell. On the human cell you have a receptor called as CCR5 receptor. I repeat, CCR5 receptor. This is the receptor for HIV virus. It means if this receptor is not there, then the HIV virus that cause AIDS cannot infect the human beings. So what we have done here is we studied about the genome, we studied the complete genome of the human being. Then what we have done, we identified which gene will produce this CCR5, and then we suppress that gene. We suppress the gene, and that human being can never get HIV, because the gate for HIV is no more. We suppressed the gene that will produce this CCR5. So it means when you understand the mechanism of infection, first I understood that the HIV virus is infecting the CCR5, then I will try to suppress that. If I do not understand the mechanism of infection, I cannot do anything about that. How to study the mechanism of infection? By studying about the genome, that is by genomics. Second application in the health, that is RT-PCR. In the context of COVID-19, you all must have heard about this RT-PCR. PCR stands for the polymerase chain reaction. Polymerase chain reaction. In this, what we do is, we take the patient sample. From the patient sample, we try to extract the DNA, and then we amplify the DNA using the PCR technique. And if you find the DNA of a tuberculosis bacterium in the human sample, think about it. Tuberculosis bacterium DNA, Mycobacterium tuberculosis bacterium DNA, you found in the human sample. Why does a human being will have the bacterial DNA? Only when that bacterium infected this human being. So you found the DNA of the tuberculosis bacterium means what? This person is infected by the tuberculosis. Okay. What is the role of genomics in this? We are finding the DNA of the tuberculosis bacterium. It means we should know about the genome of, not the human being here. We should know more about the genome of the tuberculosis bacterium, because you should be able to recognize whether it is tuberculosis bacterium DNA or some malaria parasite DNA or some coronavirus DNA. We should be able to recognize which pathogen's DNA it is. For that, you should know the sequence of that pathogen's DNA. For that, genomics is very important. So genomics plays a role in understanding the mechanism of infection and diagnosing what infection the patient is suffering from, and drug development by studying the genomics of the pathogen. It means, for example, usually we say that the one who knows us better only can cheat us better, because they know our weakness. So here what we do is, we study the genome of a pathogen, that is a bacterium or a virus. By doing that, we will understand the weakness of that pathogen, and we try to hit on its weakness. It means we try to design a medicine, a drug that can target the weak point of this pathogen. For that also, we should have the complete knowledge about the genome sequence of that pathogen, not the human being. We need to know the genome sequence of that pathogen. That is the reason why initially I said that genomics is the study of genome of either a person or an organism. We study about the genome of human being, we study the genome of the different crops for agriculture, and we study about the genomes of pathogens for the diagnosis purpose and also for understanding the mechanism of infection purpose, etc. So here, okay, we are trying to analyze the genome sequence of this pathogen, that is a bacteria or a virus. Pathogen means disease causing agent. It can be a bacteria or a pathogen or a parasite or whatever it is. Okay, so drug development. Genomics plays an important role in the drug development. Next, personal therapeutics means not a regular medicine for everybody. A separate medicine exclusively designed for you. That is called as personalized therapeutics or we can also call them as designer drugs, or we can also call the field as pharmacogenomics. Let us try to understand this term pharmacogenomics. If you observe, pharmaco is about the drugs and genomics is study about genome. So, a combination of these two is pharmacogenomics. In this, what we do is, let us suppose a patient visits a doctor. Then the doctor will read the genome sequence, the DNA sequence of the patient. By analyzing the DNA sequence of this patient, the doctor will understand what particular medicine will work for him and then will prescribe that exclusive medicine. If none of them are working for this person, then the doctor can also try to design a new medicine exclusively for this person. A new medicine that can exclusively be suitable for his genome sequence or for his DNA sequence. I repeat, a patient visits a doctor. The doctor will read the genome sequence or the DNA sequence of the patient, will try to understand which medicine will work for him the best and will prescribe that medicine. If none of the available medicines are suitable to this person, you can also design an exclusive drug for this person. That is called as pharmacogenomics or personalized therapeutics, designer drugs or pharmacogenomics. Next one, preventive care. Nutritional genomics, quite interesting. Here what we do? Let us suppose a patient goes, he is suffering from diabetes, some fatty liver, whatnot, several problems. And the nutritionist, he is visiting a nutritionist. The nutritionist will read the DNA sequence of the person. Say, I visited a nutritionist. The nutritionist will read the DNA sequence of my cell, my genome sequence, and will suggest which food is suitable to me. Not in general nutritious food, not in general healthy food. The food that is exclusively suitable for me. How did she know about it? By reading the DNA sequence of my cells. That is a quite latest field, that is nutritional genomics. This is about personalized nutrition, not in general nutritious food, not in general healthy food. The food that is particularly suitable to you according to your DNA sequence. How is that suggested? By analyzing the genomics. Then, we also use genomics in tracking the new variants of the pathogens. I hope you all remember during the COVID-19 first, second and third waves, we heard about different new variants of the coronavirus like the alpha variant, beta variant, delta variant, omicron variant, etc. Recently, there were two flirt variants. What is flirt? The name of the new variants, okay, were also discovered. Then, how to know whether it is a new variant or the same variant as the previous one? I mean, how to know whether it is a new variant called Omicron or is it the same Delta variant? By analyzing the DNA sequence. Let us suppose, okay, if the new virus DNA is different from the previous virus, if the new virus DNA is very different from the previous virus, then we declare it as a new variant. If the new virus DNA is mostly similar to the previous one, we say that it is not a new variant at all. So, we need to constantly keep a track of the evolution of the new variants, because when the new variants come, maybe we need new vaccines, or maybe we need new medicines, or maybe we need new diagnostic tools. So we should keep a track of the new variants of certain viruses or the pathogens. For that, we have something called as INSACOG. It is a consortium of several laboratories to keep tracking the new variants of the coronavirus, that is SARS-CoV-2 new variants, will be tracked by INSACOG, a consortium of several laboratories. Same way, GSED will be tracking the new variants of flu virus. So, this is another application of genomics in the health sector. Next, better understanding of MDR. MDR means multiple drug resistant. Multiple drug resistant pathogens. We also call them as superbugs. We have antibiotics that can kill the bacteria. But if the bacteria become resistant to these antibiotics, then our antibiotics will become useless. Means the bacterium is having an upper hand on our antibiotics. So we call them as superbugs. So using genome studies, we can also better understand about the multiple drug resistant strains of the pathogens. Okay, how do we know? By reading the DNA sequence. So the summary of all these applications is, when you read the DNA sequence of an organism, you will understand everything about that organism. All applications are based on that principle, right? Lastly, by knowing the DNA sequence of a human being, we understand better about our genes. When you know better about our genes, you can also do new techniques like gene therapy. Some new techniques like Casgevy for the treatment of hemophilia, thalassemia, sickle cell anemia will be possible only when you have complete knowledge on the genome. Simple. Gene therapy is possible only when you have complete knowledge about our genome. These are the applications of genomics in the health sector. Okay, we understood that it is quite important to study about the genomes. So what are the initiatives taken in India to study about the genomes? First one is IndiGen project. IndiGen project is a project. Project means a task to be completed. The task here is to study the genomes of the Indians, not the world populations, particularly to study the genomes of the Indians. This project was taken by the Institute of Genomics and Integrative Biology, IGBI, Delhi, Center for Cellular and Molecular Biology, CCMB in Hyderabad. The second project is Genome India project. What is this project? Here also it is a task to be completed. The task is sequencing 10,000 genomes. I mean sequencing the genomes of 10,000 Indians. Sequencing the genomes, I mean reading the DNA sequence of 10,000 human beings from India. And the project was launched in the year 2020. By doing this, we can better understand about the genomes of Indians, so we can develop new techniques like gene therapy which are more suitable for the Indian population. That is about Genome India initiative. Next, the genomics means studying about the genomes for understanding the rare diseases. We have to study the genomics to understand the rare diseases. Then, Indian Cancer Genome Atlas. Indian Cancer Genome Atlas. First, we should understand that cancer is a genetic disease. Means cancer is caused because of certain mutations in certain genes. So, we should understand what kind of genes will cause cancer, then we can try to develop gene therapies for cancer. I repeat, first, understanding that cancer is a genetic disease. Understanding that cancer is caused by mutations in certain genes. Then we should read the DNA sequence, that is understand about our genome, try to find out which gene is causing the cancer. Then what we can do, we can devise a new technique for the gene therapy for cancer. So what is this Indian Cancer Genome Atlas? The atlas collects biological and clinical data from the consenting patients. For example, there is a cancer hospital. 10,000 patients visited the hospital, and the hospital management wanted the blood samples from all the patients. Most of them refused. They said, we will not give the samples for the study. Some 1,000 people given the consent. From that consenting patients take the samples and do the genomic study. All these are cancer patients and you are collecting their genomes and studying. So you will understand what genes caused cancer in them, what is common in all of them. Okay, what genes caused cancer in them, that is possible. Okay, and a compilation of the genomes of all these cancer patients. I repeat, the compilation of the database of the genomes of all these cancer patients is called as Indian Cancer Genome Atlas. Lastly, Human Microbiome Project. Microbiome. I told you that genome is a set of genes, genetic material. So same way, biome is a set of biological organisms where inside the human being, what are they? The friendly bacteria. The friendly bacteria. We have a lot of bacteria, some good bacteria and some bad bacteria inside the human body. Not only inside on the surfaces also, hands, skin, digestive system, that is the large intestine, small intestine. And these bacteria, interestingly, in our body, we have more bacterial cells than the human cells. In the human body, we have more bacterial cells than the human cells. So it is not surprising that these bacteria will have enormous impact on our life, our digestive capacity, okay, and our moods, our vitamin production, whatnot. These bacteria will play important role in several aspects. The rate of our metabolism, our digestion capacity, how efficiently we can produce vitamins and how healthy we are, how is our immune system, how are our moods, are we a pleasant human being or a stressed person. All these are influenced by this human microbiome. Microbiome, that is a total set of all microorganisms living inside the human body. So this is about studying the DNA sequence of not human beings, studying the DNA sequence of those microbes present inside the human being. Human microbiome genomics, that is another application. And we can also do genomics for the understanding of neonatal disorders. Neonatal, neo means new, natal means birth. So neonatal means newborn. It means we have to collect the DNA samples from all the newborns and do the genomics for the understanding of neonatal disorders. Then after that, National Initiative on Climate Resilient Agriculture. It means, okay, climate resilient agriculture. Resilient means what? Tolerant or resistant. Resistant to what? Resistant to climate. Means we are speaking about some drought resistant or temperature resistant or light resistant crops. Okay. National Initiative on Climate Resilient Agriculture. That agriculture that is producing those crops which are resilient to the climatic changes. To study about them, there is one national initiative. Next, genomics assisted breeding in the crops. It means study the genomes of the crops and with the knowledge of the DNA sequence, do the selective breeding with the best varieties. How to know which are the best varieties of the crops? By analyzing their DNA sequences, by analyzing their genome sequences. Next, National Bovine Genome Center for Indigenous Breeds. This is to study about the genomes of the cattle, cows, buffaloes, which are the indigenous varieties in India. By studying the genome sequences of these bovine organisms, we can try to save them. We can try to save our indigenous varieties. If you want to save a particular variety also, you should know about its genome sequence. Lastly, okay, Indigo chip. India information about the indigenous cattle is collected. It means all indigenous variety cattle, cows, buffaloes, indigenous varieties. Their DNA is collected, I mean their blood is collected. From the blood they extract the DNA. Through these blood tests, what they do, they store the DNA sequences of all the indigenous varieties of the cattle in a database. That database is called as Indigo chip. I repeat, there are a few indigenous variety of the cattle like cows, buffaloes, etc. We take their blood samples, extract the DNA and read the DNA sequence, that is know the genome sequence, and create a database of these genome sequences. That database is called as Indigo chip. So finally the summary is, if you want to create new varieties or track the new variants or diagnose an organism, or you want to do conservation, you want to know anything about an organism, you have to read its DNA sequence. That is how the genomics will have its role in the biotechnology applications in all the fields. Now, conclusion for this answer. Genomics has come to touch every sphere of human life like health, agriculture, animal breeding, and playing a vital role in the advancements in biotechnology. That's all. With this we completed our second topic, that is genomics and its applications in various fields. The third topic we are going to discuss today is Agni missile with MIRV technology. Agni missile with MIRV technology. Defense Research and Development Organization DRDO conducted the first successful flight test completed. This is the flight test of indigenously developed Agni 5 missile with multiple independently targetable re-entry vehicle. First, what is this Agni 5? Basically, Agni 5 is a ballistic missile. Ballistic missile is a missile that will travel in a parabolic path. If any missile travels in a parabolic path, we call this as ballistic missile. And to be more specific, we call this as ICBM, means Intercontinental Ballistic Missile. Reason is, it has a range of 5,000 kilometers. If this is the launch site, the target site can be 5,000 kilometers away. 5,000 kilometers means it can hit a target in a different continent also. So we call this as Intercontinental Ballistic Missile, ICBM. And this Agni 5 is not only an intercontinental ballistic missile, it is also having a new capacity, that is multiple independently targetable re-entry vehicle. Means what this Agni 5 can do is, if you launch Agni 5 here, it will hit one target, then again it will hit another target, and then it will hit another target. Like this it can hit 100 targets. Like this it can hit 100 targets. Not only that, it can hit 100 targets. After hitting them all, it can come back to the launch site, hit all the targets, and come back to the launch site. That is the meaning of multiple, because it can hit multiple targets and it can independently target them. It can independently target all those 100 sites. After hitting them all, it can also make a re-entry to the launch site. So the pattern is like this. It will hit all the targets and it will come back to the launch site. So we say that Agni 5 is an intercontinental ballistic missile with multiple independently targetable re-entry technology. This is a new technology. And it was recently flight tested. The test name is known as, the flight test is named as Mission Divyastra. The flight test is named as, the missile is Agni 5, its test is called as Mission Divyastra. It was carried from APJ Abdul Kalam Island in Odisha. Various telemetry and radar stations are tracking and monitoring this Agni 5. Various radars and tracking stations are tracking and monitoring this Agni 5. And it is a 3-stage surface to surface missile. 3-stage means what? As we also study in the space technology, any rocket or a missile launch vehicle will have different stages. So, we ignite the stage 1 at the launch. Once the stage 1 is fully burnt, it will ignite the stage 2. Once the stage 2 is fully exhausted, fully burnt, then it will ignite the stage 3. So this is how the different stages work. We have different vehicles like 3-stage vehicles, 4-stage vehicles like that, but okay, this is a 3-stage vehicle. Okay, it is a 3-stage vehicle and also it is a surface to surface missile. Surface to surface missile, if you observe here, it is starting here and it is hitting the target here. This is one surface and this is the other surface. Means these are surface to surface missiles. Then what are the other missiles? For example, if you launch this here and it means you are launching it from an aircraft, and if it hits a target on a surface like a ship or a military tank, etc., then it is called as air to surface. Air to surface. But if a missile is launched on the surface and if it is hitting an aerial threat, if it is hitting an aerial threat and destroying that enemy's missile while it is in the air, then we call it as surface to air. Now you decide whether Agni 5 is air to surface or surface to air or surface to surface. Agni 5 is a 3-stage surface to surface solid propellant. It means we use solid fuel while launching this Agni 5, not the liquid fuel, not the cryogenic, not the semi-cryogenic fuels. We use the fuel which is in the solid state. And as we already discussed, its range is 5,000 kilometers. So we call this as intercontinental ballistic missile. Okay, and it can carry a payload of 1.5 ton warhead. In the space technology, payload means satellites or instruments. In the defense technology, payload means the munition. That means the explosives. That is the warhead. It can carry 1.5 ton warhead. Okay. A MIRV is a ballistic missile designed for the exoatmospheric deployment. Exoatmosphere means It will travel so high that it is exoatmosphere. It means it went to such a high altitude that it is in the outer layers of our atmosphere, not in the inner layers of our atmosphere. So it is exoatmospheric. Housing multiple warheads. Why do you want multiple warheads? Because it is multiple independently targetable re-entry vehicle. It can hit several targets. To hit several targets, it needs several warheads also. So, each warhead is capable of targeting a distinct target hundreds of kilometers apart. It means, okay, between these two, you can have more than 100-kilometer distance. It means those independent targets are also not near to each other. It can hit a target here and again travel for 100 kilometers and hit one more target. Again travel for 200 more kilometers and hit one more target. And like that, it can hit 100 targets which are hundreds of kilometers away from each other. After doing all this work, it can come back sincerely to its launch site. That is a super capability of Agni 5. While primarily associated with intercontinental ballistic missiles equipped with thermonuclear warhead, it means Agni 5 is equipped with thermonuclear warhead. It means not only the classical explosives, it can also use nuclear explosives. And this concept extends to intermediate variant. Look at this word intermediate variant and try to think about it. In this term intermediate variant, are we discussing about Agni 5? No. We are discussing about the other missiles which have slightly lesser capability than Agni 5. So what are they? They are MRVs. Okay, observe these two. What is MIRV? This is multiple independently targetable re-entry vehicle. So MIRV. This is about Agni 5. But we have some intermediate variants. Means some variants which are better than the regular ballistic missiles but inferior to MIRV Agni 5. So what is this? We call them as MRVs, not MIRV. This is MRV. MRV means it is multiple re-entry vehicle. Multiple re-entry vehicle. Multiple target re-entry vehicle. If you observe, what is the difference between these two? Just the word independent is missing. It means MRVs can hit multiple targets. MRVs can hit multiple targets but not independently. Okay, no independently aiming. With this, what is the problem? They may not be able to precisely hit the target because they are not targeting each object. For example, they want to hit this Agni 5 want to hit some 100 different military tanks and it is not independently aiming at each of them. So there is no guarantee that it will hit the target. So MIRV is multiple independently targetable. It means each target is independently aimed. Whereas MRV is multiple re-entry vehicle. It will hit multiple targets but not as precise as the MIRV. These are intermediate variants. Okay. Then advantages of MIRV. It can release multiple warheads and thereby reduces the number of missiles and launch facilities. For example, one Agni 5 can hit 100 targets. It means you have saved the energy of 99 missiles. One missile can hit 100 targets. It means you need not launch 100 missiles. Lot of fuel, energy, economically better. Okay. With MIRV, the effectiveness of an anti-ballistic missile system. Anti-ballistic missile system. If we have a ballistic missile, our enemies should have an anti-ballistic missile. Right. If I have a missile, then the enemies should have the anti-ballistic missile defense. Same way, if they have a ballistic missile, then we should have anti-ballistic missile system. Like for example, we have anti-ballistic missile system 5-layer protection on Delhi. Same way, in Israel also we have multi-layer anti-ballistic defense system. It means if enemies launch a ballistic missile, they have a defense system. If you have this MIRV, enemies missile defense system, anti-ballistic missile system will become less effective. It means they cannot destroy our Agni 5. I repeat, if we have ballistic missiles, the enemies should have the anti-ballistic defense system. If we are launching the Agni 5, then enemy's anti-ballistic defense system cannot stop it. So it will make their defense system less effective. This is another reason why we need Agni 5. Okay, this is about Agni 5. Now, what other countries have this MIRV technology? USA, UK, France, China, Russia, Pakistan. Because these countries have this MIRV technology, particularly because China and Pakistan is having this MIRV technology, it is important for India also to have this MIRV technology when you try to see this in the geopolitical context. Then, some key features of Agni 5. Its weight is 50,000 kilograms, 1.75 meters height, 2 meters diameter, and it can carry 1650 kilograms of warhead, and it can at a time carry 2 to 12 warheads at a time, and its speed is 25 Mach. It means it can travel at a speed of 25 times the speed of the sound. It means it is a hypersonic. Okay, so it is a hypersonic intercontinental ballistic missile with MIRV technology with nuclear capabilities. That is a speciality of Agni 5. And it can travel at a speed of 8.5 km per second or 29,401 kilometers in 1 hour. It is equipped with advanced navigation systems, including a ring laser gyroscope inertial navigation system called as NAVIC. Means it is having advanced navigation system. Navigation system will give the route map. It means because it is having the advanced navigation system called as NAVIC, it can precisely hit the target. It can correctly go to the target and hit that. Launch flexibility from mobile launcher. It means you can also launch Agni from a moving launcher. The launcher can be mobile also. Still the Agni 5 can be launched and it will correctly, precisely hit the target. Next, Agni 5 trajectory and the thermal resilience. Look at this word, thermal resilience. We have seen already that it will go into the exo-atmosphere. When it goes into the exo-atmosphere, it should come back into the atmosphere, into the Earth's atmosphere. Is it clear? Like this, it will go into the exo-atmosphere and here it is making entry into the Earth's atmosphere. In the Earth's atmosphere, there is more air resistance. We have more air resistance. This air resistance will create friction, and the friction will release heat. So therefore, this Agni 5 is not only going and hitting the other target, and it should also come back to the launch site on the Earth. So therefore, it should have a thermal resilience. It means heat resistant. While it is entering back into the Earth's atmosphere, it will experience a temperature of 4000 degrees Celsius because of this friction. Why so much of friction? Because it is a hypersonic missile, 25 times the speed of the sound. When it is entering into the atmosphere at such a high speed, it will with a high speed it will cross the atmosphere and creates enormous friction. So the temperatures will go up to 4000 degrees Celsius. But still, the temperature of the heat shield, Agni 5's heat shield, will maintain Agni 5 temperature to be only below 50 degrees Celsius. Means Agni 5 is not heating up to 4000 degrees Celsius. Means it will not be burnt. You can reuse it again. That is about the thermal resilience that it is having. That's it. That is about our third topic, that is the Agni 5. Moving on to the fourth topic, that is sodium ion batteries. So what is a sodium ion battery? How does it work? Simple. It works similar to the lithium ion batteries. It works similar to the lithium ion batteries. But with lithium ion batteries, we have some limitations and some problems associated with them. So when compared to the lithium ion batteries, sodium ion batteries are better. So we are trying to develop them. And basically, what are these lithium ion batteries or the sodium ion batteries? These are all rechargeable batteries. And where do we use these rechargeable batteries? We use them in our mobile phones or laptops or tablets or electrical vehicles, EVs, and many other applications. Wherever you are using a rechargeable battery, mostly it is a lithium ion battery. But today we are discussing about sodium ion batteries. How does these sodium ion batteries work? See, this is, if you can see, these are the sodium ions. Is it clear? And this is anode. And this is cathode. These are the sodium ions. And this is a membrane. This is a membrane or a separator. Then what happens is, during charging, it means when you charge the battery, the sodium ions will move in this direction. That is from the cathode to the anode. Then the battery will get charged. During discharge, means when you are using it, when you are using your mobile phone or laptop or electrical vehicle, then the sodium ions will move in this direction. That is from the anode to the cathode. Then the battery will be discharged. You should again charge it. Then the sodium ions will travel back from the cathode to the anode. This is how the sodium ion batteries work while they are getting charged or when they are getting discharged. And if it is lithium ion batteries, same technology. But if it is a lithium ion battery, then instead of sodium ions, the lithium ions will travel between the cathode and the anode. That is the only difference between the sodium ion batteries and the lithium ion batteries. Now, let us see some introduction about these sodium ion batteries. University of Houston, scientists developed an electrolyte that contributes significantly in making the sodium ion batteries more commercially viable. For any batteries, the cost of the battery should be commercially viable. So these scientists have developed a new electrolyte. With the help of this new electrolyte, we can make cheaper sodium ion batteries. Okay, what is an electrolyte? If you observe, we have seen that the sodium ions are moving, okay, from the cathode to the anode and from the anode to the cathode. For these sodium ions to move through this separator, we need a medium. The medium for the sodium ions to travel is called as an electrolyte. We can use different material as electrolytes, but the scientists in the University of Houston developed a new electrolyte which is cheaper. So with this new electrolyte, we can make the sodium ion batteries at a lesser cost. This is a small news item related to the sodium ion batteries. Sodium based battery technology might soon be available as an alternative to the lithium based ones. Sodium ion batteries are rechargeable batteries which require sodium ion movement between the electrodes. We have already understood. We need sodium ion movement between cathode and anode during the charging and discharging of the battery. The cathode for these batteries is manufactured from sodium. Okay, it is called sodium ion battery because of two reasons. Why this is called sodium ion battery? Because the cathode material is sodium and also the ions traveling are also sodium ions. That is the reason why it is called sodium ion battery. Cathode is sodium and the ions traveling between the cathode and anode are also sodium ions. Then, why do we require sodium ion batteries? Because we have some problems or issues with the lithium ion batteries. Let us try to understand what are the issues with the lithium ion batteries. Lithium ion extraction led to environment harming mining process. First, it means the mining of lithium ions or the mining of lithium, okay, is harming the environment. I repeat, the mining process of lithium is harming the environment. Then next problem. It is non-reusable as recycling of lithium is very expensive. It means maybe with great difficulty you can recycle the lithium, but it is too costly that it is not commercially viable. So you cannot reuse the lithium ion batteries. Third problem, limited availability of lithium. You all know that in India we do not have rich resources of lithium. So firstly we need to import. Okay, we can import, but it is also not available in many countries so that we have a choice of from which country to import. It is particularly available only in the 56% of the world's lithium supply comes only from the lithium triangle countries called as Bolivia, Argentina and Chile. It means most of the world countries should import lithium from only these three countries, means increasing the demand. So making it costly, not only making it costly, availability also matters. So we have seen three issues related to the lithium ion. First one, the mining process of lithium is harmful to the environment. Second one, it is not reusable because recycling the lithium is very costly that it is not commercially viable. Third one, lithium is having a limited availability, 56% of the total lithium in the world coming from only 3 countries increasing its demand and also the cost. Next, this is the reason why we need sodium ion batteries. And what are the benefits of sodium ion batteries? Sodium ion batteries are cheaper. Why? Because sodium is more available than the lithium. Also not available only in certain countries. Okay, next, non-flammable. You might have heard about such blasting or accidents of this electrical vehicles, particularly two-wheelers and the other vehicles. It means lithium ion batteries are more flammable. Sodium ion batteries are non-flammable and operate well in the colder temperatures also. They are less likely to experience thermal runaway, which is a condition common in the lithium ion batteries. Lithium ion batteries are experiencing the thermal runaway. That is the reason why we heard about some electrical vehicles catching the fire, especially in the summer seasons. That problem will not be there with the sodium ion batteries. Next, sodium ions move from the negative electrode to the positive electrode during discharge and back when they are charging. The battery consists of an electrolyte that allows the movement of these ions. Okay, so with this, what all we understood? What are sodium ion batteries? We understood what are the sodium ion batteries here. Then we understood that they are similar to the lithium ion batteries in the working mechanism. And then we discussed about the limitations of the lithium ion batteries. Then the advantages of sodium ion batteries. And here is the comparison between sodium ion batteries and the lithium ion batteries. Let us see that comparison. These are safer as they do not explode or catch fire. And these are more prone to catching fire, lithium ion batteries. Next, they have shorter lifespan and can be recharged fewer times compared to the lithium ion batteries. Okay, in the first parameter, which is better? In the first parameter, which is better? Here we can easily see that sodium ion batteries are better. But if you observe, sodium ion batteries have a lesser lifespan. It means if you are using it in an electrical vehicle, you can charge that electrical vehicle only few times, not as many times as you can charge if you are using the lithium ion batteries. So in this matter, lithium ion batteries are better. Next, they have lower performance compared to lithium. They have better lithium ion batteries have better performance. So in the performance aspect, the lithium ion batteries are better. Next, environmental impact. We have already seen that the mining process of lithium is harmful to the environment, so sodium ion batteries are more environmental friendly. So in the environment aspect, sodium ion batteries are better. Next, energy density. It means they have lower energy density compared to lithium ion batteries. It means with this the battery size increases, but here higher energy density. It means in a small battery can give more mileage or more hours for working. For example, if you charge the cell phone, if the cell phone is using lithium ion battery, then you can use the cell phone for a more duration with one charge. If you are using it in an electrical vehicle, you can it can give you more mileage, more distance can be covered in one charging. Okay, smaller battery can hold more electricity. That is about the higher energy density. Smaller battery, smaller surface area, okay, can store more energy. Okay, so in this aspect, lithium ion batteries are better. Next, sodium ion batteries are cheaper. So in that aspect, sodium ion batteries are better. Next, okay, let us stop the discussion about which batteries are better, and the last point is about what batteries are suitable for what kind of situations. For example, sodium ion batteries are suitable for large scale energy storage applications. I mean, when you do not have the space constraint, a very huge battery is okay, but you want to store energy at a cheaper cost, then this is better. If the size matters, it means if you want a slim and sleek cell phone, if you want a smaller electrical vehicle, then sodium ion battery is not suitable. When size does not matter, a large battery is okay, but you want it at a cheaper cost, you want to be more safer and you want to be more environmental friendly, then it is sodium ion battery. But when the size matters, then it is lithium ion battery. So it is also not very appropriate to compare which batteries are better. It is also about in what situation we use which battery. Okay. And finally, sodium ion batteries are slowly finding their way. You can consider this as a concluding statements. Sodium ion batteries are slowly finding their way into the electrical vehicles. It means we started using them in the electrical vehicles also. Okay. And because the lithium ion batteries became very costly, environmentally harmful, and thermal accidents, heat accidents, fire accidents, etc. Moreover, sodium ion batteries address the concerns of geopolitical and supply chain issues. Geopolitical means our relation with the lithium triangle countries like Chile, Bolivia, Argentina. Geopolitical issues will not be there if you are using the sodium ion batteries. And the material which will be used in such sodium ion batteries can be fabricated in-house in India. We need not import sodium. So it means sodium ion batteries are the suitable example for make in India and make for India. So this is the reason why India needs the sodium ion batteries. That's all about the sodium ion batteries. Now quickly moving on to the fifth and the last topic in today's session, that is gene therapy and the applications of gene therapy. Let us try to understand what is gene therapy and what are the applications. Gene therapy is a DNA based technique that aims to fix a faulty gene. I mean, if you have a mutated gene, then this mutated gene is causing a disease. Then what you have to do? Correct that mutation or correct that error. What is a mutation? Mutation is a mistake or an error in a gene sequence. Correct that error so that the disease can be cured or treated. Because you are giving the treatment, we call it as a therapy, and because you are giving the treatment using the gene, we call it as gene therapy. We have two types of gene therapy, somatic gene therapy and embryonic gene therapy. But before discussing the types of gene therapy, let us try to understand the mechanism of gene therapy. For gene therapy, we are using gene editing tool. Gene editing tool like CRISPR-Cas9. So let us try to understand how the CRISPR-Cas9 works. CRISPR-Cas9 cuts the mutated gene, mutated or faulty gene, and adds the correct gene in the gap. So simple, two steps. It will cut or delete the unwanted gene, mutated gene which is causing the disease, and it will add the correct gene so that the disease can be treated. Let us try to understand, okay, how the CRISPR-Cas9 is doing it. In the CRISPR-Cas9, CRISPR stands for, what is CRISPR? This is CRISPR. CRISPR is a guider RNA. Guider RNA means what? An RNA that guides. An RNA that guides. Okay. Whom does it guide? Whom does this CRISPR guide? CRISPR is guiding. It guides Cas9. Okay, what is Cas9? Cas9 is here. This is a nuclease enzyme. Nuclease enzyme. Nuclease means what? If you observe, the word nucle stands for nucleic acid. Which nucleic acid we are discussing about? DNA. So the word nucle means nucleic acid, that is DNA, plus ase. The word ase means cut. So club the two words. Nuclease. Nuclease is an enzyme that can cut the DNA. It is an enzyme that can cut the DNA, that is a nuclease. Means this is like a scissor. It can cut the DNA, but the scissor does not know where to cut. Cas9 is a scissor. It can cut the DNA, but it does not know where to cut. So an RNA called CRISPR is guiding the Cas9 about where to cut. Let us see how does it guide. Before that, look at this. This is a double-stranded DNA. One strand sequence is this and the other strand sequence should be opposite to it. I mean, when you have thymine here, adenine will be here. When you have adenine here, thymine, I hope you all know that adenine will always pair with thymine and guanine will always pair with the cytosine. These are the two strands of the DNA. And this is our gene. What gene it is? It is the mutated gene. Okay, this is the mutated gene. So what the CRISPR-Cas9 is supposed to do? It should cut the mutated gene and add the correct gene in that gap. Now look at this CRISPR sequence. Okay, the mutated gene is AATCT, and the CRISPR is UUAGA. From this you can understand that the CRISPR can precisely bind to this mutated gene. If you notice, this CRISPR, that is this guider DNA, cannot bind to this gene or this gene because U cannot pair with G, U cannot pair with T, and A cannot pair with G. So this RNA will bind exactly to this gene, the mutated gene, okay, and then now Cas9 is linked to the CRISPR. I repeat once again, CRISPR is a guider RNA, means it is an RNA that will guide the Cas9. It is by nature, it is an RNA. By function, what is its function? It will guide the Cas9. What is Cas9? Nuclease. It is like a scissor. It can cut the DNA, but it does not know where to cut the DNA. So CRISPR and Cas9 are linked. CRISPR will go and bind to this mutated gene. Once the CRISPR binds to the mutated gene, Cas9 will cut that gene which is bound to the CRISPR. Cas9 will cut that particular gene which is bound to the CRISPR. This is just like first mark with a chalk piece where to cut and then start cutting that. So the scissors role is played by the Cas9 and that chalk piece role is played by the CRISPR. This is the mechanism of CRISPR. So like this, if you are deleting the mutated gene and adding the correct gene in a somatic cell, if you are deleting mutated gene and adding the correct gene in a somatic cell, then it is called somatic gene therapy. Then what do you mean by a somatic cell? Somatic cell means body cell. Somatic cell means body cell, like skin cell, heart cell, liver cell, like that. Usually this is given to treat a disease. After the disease comes in the patient, to his body cells we do that. For example, if there is a skin cancer here, I will use the CRISPR-Cas9 on the skin cell. Then it is called somatic gene therapy. If there is a blood cell related problem in my body, then I will use CRISPR-Cas9 on my blood cell. Then it is called somatic gene therapy. But if you are doing this correction, I mean deleting the mutated gene and adding the correct gene in the embryonic stage, that is before the birth, we use it to prevent the disease. We use this to prevent the disease. It means gene therapy can be used either to prevent the disease or to treat the disease. For preventing, we use embryonic gene therapy. Embryo is a stage before the birth. Okay, when we do the correction at the embryonic level, we call it as embryonic gene therapy. We can even prevent the genetic diseases. Whereas if you do this correction in the somatic cells, it is called somatic gene therapy. Then this is CRISPR-Cas9 mechanism. What are the applications? One application of the gene therapy is gene silencing. Means what? You are silencing a gene. Okay. Which gene are you silencing? Oncogene. Okay. What is an oncogene? This is the gene that causes cancer. The gene that causes cancer. Oncogenes cause cancer. So if you silence the oncogene, cancer can be suppressed. Simple principle. Oncogenes cause cancer. So if you delete the oncogene, then the cancer can be suppressed. How to delete the oncogene? Use CRISPR-Cas9 to cut, I mean delete the oncogene. Oncogene is causing the cancer, so once oncogene is deleted, then the cancer can be suppressed. One example for this is BRCA1 gene. This is a gene that can cause cancer. What is this BRCA1? BRCA1 is the example of an oncogene and it is causing cancer. And delete this oncogene using the CRISPR-Cas9. I mean CRISPR will bind to BRCA1. Simple. CRISPR binds to BRCA1 and then Cas9 cuts BRCA1. With this what happened? The cancer suppressed. Cancer cells cannot further grow. This is how we can treat cancer by using CRISPR-Cas9 in the somatic cells of a cancer patient. This is one application. Another application is Cas-JV technique. One more application is Cas-JV technique. Let us see how does it work. But to understand that, we need to know a little bit more about our hemoglobin. Listen carefully. We need to know more about the hemoglobin. In hemoglobin, heme stands for iron and globin is a protein part. And this protein is having 4 parts or subunits in that. 4 parts or subunits in that, like this. And we have different types of hemoglobins. Like for example, okay, see, hemoglobins are ferroproteins. Ferroproteins means what? Iron containing proteins. Iron containing proteins. Heme is a large organic molecule having one atom of iron. Heme is a large organic molecule. It means heme is not iron. Heme is a bigger organic molecule because it is there in the living organisms. And this heme will have one atom of iron, that is heme. And the protein part is called as the globin. Globin is the protein part. This protein part will have 4 parts in it or 4 subunits in that. Or we can also say 4 polypeptide chains. I hope you all know proteins are made up of amino acids. Proteins are made up of amino acids. And the bond between 2 amino acids is called as a peptide bond. Therefore, proteins are called as polypeptides. Proteins are the chains of amino acids and we also call them as polypeptides. One globin will have 4 subunits or 4 parts or the 4 polypeptide chains. And we have different types of hemoglobins. For example, okay, in the HbA, what is HbA? This is adult hemoglobin. Adult hemoglobin will have alpha to beta 2. Alpha, alpha, beta, beta. Then it is called HbA, that is adult hemoglobin. Is it clear? But if it is having, okay, other subunits like alpha to gamma 2, that is alpha, alpha, gamma, gamma, then it is called fetal hemoglobin. This is called as fetal hemoglobin. Now, we have some abnormal hemoglobins also. One is hemoglobin S that causes sickle cell anemia. This adult hemoglobin and the fetal hemoglobin both are at different stages of the human life. As an adult, I will have adult hemoglobin, but a fetus. Fetus means what? The baby still inside the womb of the mother. The fetus will have fetal hemoglobin, but they both are normal. But we have some abnormal hemoglobins also. One example for abnormal hemoglobin is hemoglobin S that causes sickle cell anemia. What causes sickle cell anemia? Okay, let me explain this. Okay, let us suppose that you have beta chain. I told you that proteins are the chain of amino acids. In the beta chain, you have several amino acids. What are all these? These are the amino acids. Amino acids in what? In the beta chain. Where is beta chain? I told you adult hemoglobin is alpha, alpha, beta, beta. So we are discussing about that beta chain in the adult hemoglobin. HbA, adult hemoglobin. You have 4 subunits, 2 alphas, 2 betas. Now we are speaking about that beta subunit. Each subunit is made up of several amino acids. In that 1, 2, 3, 4, 5, 6. The sixth one is originally glutamic acid. Okay, this is the original amino acid. Original amino acid. What is glutamic acid? It is the name of an amino acid. It is the original amino acid at the 6th position in the beta chain of a hemoglobin. If this glutamic acid changes into valine, then it causes sickle cell anemia. It causes sickle cell anemia. Here the RBCs will become sickle shaped, elongated like this. Okay, so that they cannot pass through the blood capillaries. This is a blood capillary. Blood capillary is very thin here. and the RBC became very elongated, sickle shaped, so it cannot pass through the blood capillary. So with severe bloodlessness, obstructions in the blood flow, the person will die. That is what happens when the glutamic acid is replaced by valine. But if glutamic acid is replaced by lysine, then it will form HbC. This is another defective hemoglobin. This is also one abnormal hemoglobin. This will also cause anemia, but it is not called as sickle cell anemia. These are HbS and HbC are the two abnormal hemoglobins. And you might have heard about thalassemia also. What is the difference between sickle cell anemia and thalassemia? In sickle cell anemia, you have a defective hemoglobin, but thalassemia is about absence of hemoglobin. It means if hemoglobin is completely absent, then we call it as thalassemia. Means, okay, for example, this thalassemia is two types. If, okay, look at here, in this, like alpha subunits are absent. It means you have only beta beta, alpha missing. Then it is called alpha thalassemia. Then, okay, in this original hemoglobin, alpha alpha beta beta, beta missing, you will have only alpha alpha. Then it is called beta thalassemia. So, sickle cell anemia is a defective beta chain, whereas beta thalassemia is when the beta chain is totally missing, not a defective beta chain. Beta chain is completely missing. So in thalassemia, there will be absence of hemoglobin. In sickle cell anemia and HbC, there will be defective hemoglobins. Now, how to do gene therapy? Okay, how to do gene therapy for this thalassemia and sickle cell anemia? Let us try to understand this new technique called Casgevy. This is the last topic in today's session. Let us try to understand Casgevy. Okay, see, as a fetus, you have fetal hemoglobin, that is HbF. As an adult, you have HbA. But if this adult is suffering from sickle cell anemia or thalassemia, etc., then HbA is non-functional. Is it clear? I repeat, an adult is supposed to have HbA, but if this adult is suffering from sickle cell anemia or thalassemia, then HbA, the adult hemoglobin, is missing. So now think logically. Which is better? Is it better to not have a hemoglobin at all? Or is it better to start producing fetal hemoglobin? You are an adult, but okay, what if you start producing? You start producing HbF, fetal hemoglobin, that you were producing when you were in the mother's womb. Then you will have something better than nothing, isn't it? Something better than nothing. It means here, due to sickle cell anemia or thalassemia, anyway you don't have the adult hemoglobin. And because you are adult, you don't have adult hemoglobin. So what if you start producing fetal hemoglobin? I repeat, due to sickle cell anemia, thalassemia, you will not have the normal adult hemoglobin. Because you are an adult, you stopped producing the fetal hemoglobin. Now the solution given by Casgevy technique is start producing the fetal hemoglobin. For example, I am a sickle cell anemia patient. I don't have adult hemoglobin. If I start producing the fetal hemoglobin, then I will have some or the other hemoglobin. That kind of technique is called as Casgevy technique. In Casgevy technique, what we do is we delete this BCL11A gene. What this gene will do? This gene will prevent the production of fetal hemoglobin. I repeat, BCL11A gene prevent the production of the fetal hemoglobin. What you do? Use CRISPR-Cas9. Use CRISPR-Cas9 and delete the BCL11A gene. So, I repeat here, BCL11A gene will prevent the production of fetal hemoglobin in the adult. I am an adult. I don't have the fetal hemoglobin because of the BCL11A gene that is preventing the production of fetal hemoglobin in my body. So what I will do, I will use CRISPR-Cas9 to remove that BCL11A gene. Now nobody to prevent the fetal hemoglobin production in my body. I will produce the fetal hemoglobin. Then I will have at least fetal hemoglobin when I don't have the functional adult hemoglobin because of the sickle cell anemia. This is about the Casgevy technique. These are some applications of gene therapy in the matters of health. These are the 5 topics that I wanted to discuss as a part of the first session in our Conquer Prelims program. That's all for today. Thank you so much. If the session is of some help to you, do share your comments. Thank you.